Hi everyone, my name is Edgar Ferrer Gonzales and I'm a PhD student at Rutgers University in the Cellular Molecular Pharmacology program. I come from the small island of Puerto Rico where I started my journey as a STEM professional. I always had a passion for science and the understanding of how nature works. During my time at the University of Puerto Rico at Mayagüez, I had the great opportunity to do scientific research fairly early on. At first, I was developing methods to grow bacteria in laboratory that are typically considered uncultivable, meaning that they do not grow in the lab. And as I continued to grow in this field, I started to develop more interest on what would be the medical and industrial applications from these uncultivable bacteria. Eventually, my desire for a medical application won me over, and I decided to pursue my doctoral studies at Rutgers University in the biomedical science field. Here, my research is focused on the development and validation of new antimicrobial targeting drugs. We focus particularly on drug-resistant infections, meaning bacteria that are actually resistant to our current standard of care. Normally, we live in a symbiotic relationship with a diverse microbial community that encompasses various bacteria species. These microorganisms supply our body with essential nutrients and to some extent, they protect us from bacterial infections. However, we might develop a bacterial infection when the balance within our community gets disrupted. A microorganism might have entered through an open wound. We could have ingested contaminated food or exchanged bodily fluid with an infected human. If left untreated, bacterial infection can cause severe damage to our bodies, and in some cases, even death. Therefore, the discovery of antibiotics back in 1928 is considered one of the most important discoveries of medical science throughout human history. Unfortunately, we have abused of these life-saving drugs for so many years, and as a result, we are now facing a growing epidemic of drug-resistant superbugs, of which many have already developed resistance to all of our current antibiotic drugs available. To address this problem, my research project focused on testing various small molecules across multiple drug-resistant pathogens and identify what cellular process they target. Currently, our most successful candidate has been shown that it inhibits the coordination of the bacterial cell division process by targeting a massive regulatory protein that recruits all the proteins involved in cell division. More importantly, we found that by altering the state of this protein with our drugs, certain bacterial pathogens, such as MRSA, become susceptible to drugs which is already resistant to. This is just the beginning of my exciting career as a scientist, and I hope that eventually these findings could save millions of lives. During this pandemic, I have faced multiple challenges since most of my research involves the use of core facilities or collaborating with other researchers. As a result, most of the hands-on research has become lengthier than normal, but this is because we must maintain a safe environment for everyone. Besides this, I also started to work more remotely from home, and this also gives me the opportunity to spend more time with my loved ones and actually focus on other stuff besides my research. I hope you guys enjoyed this video.